Hey everybody, so I'm going to go through the test 3 review here today. I have had the test in front of me as I made this, so everything that is covered on the test will be mentioned in this video, particularly in chapter 9, which we'll see here in a second. I have outlined the specific definitions that you need to focus on. I did that since chapter 9 was kind of thrown at you, and actually one of the classes only had really a day to learn chapter 9. So focus on what I put here. If there are things that are confusing, please ask me any questions. You can also email me. But this should summarize everything you should be looking at. So the first thing that we had covered with this unit was the different experiments throughout the history of DNA. And so the first one we talked about was Griffith's experiment. One thing I did want to note is that the expectations as far as how you study these different experiments. So you should be able to recognize each of their names and tell me who did what. So you should understand the basis of all of their experiments and be able to say, so-and-so made X conclusion. Use the in-class chapter questions that we did to kind of help you quiz yourself and understand what those kind of questions could look like. But the first scientist that we talked about was Griffith, and so he worked with two different forms of bacteria. He worked with a S form that was deadly, and he worked with an R form that was non-deadly. And so what he was doing was injecting mice with these bacteria. And so when he would inject mice with the live S form of the bacteria, they would all die. When he would inject the mice with all of the live R bacteria, they would live. So the kind of big conclusion that he was able to get came from the last kind of portion of his experiment, which is this point right here. So what he did was he heat killed that S form bacteria and combined it with a live R bacteria. So what we should be thinking is, okay, well, the deadly form is killed, so that shouldn't have an effect. And the form that is alive is that R non-deadly form. So what we would expect is that the mice would live after being injected with this combination. But what he actually saw was that all of the mice died. And so based on this, he concluded that something from that S deadly form of the bacteria must have transformed in the R form of the bacteria to make the R form deadly. So he identified a transforming principle, and that was kind of his overarching conclusion. Something was transforming in this process. And then we get into Avery. So Avery wanted to identify what is that transforming principle. What is the thing that is being passed from the deadly S bacteria into the non-deadly R bacteria to make them deadly? And so he did 3 different tests. They're outlined further in your notes, but he did a qualitative test, chemical tests, and enzyme tests, and all of these concluded that DNA was that transforming principle. So everything lined up. It all matched with DNA. However, there were some critics who were not convinced that DNA was actually the culprit. They wondered if maybe proteins could be what is causing this transformation. So Hershey and Chase came in and used bacteriophages, which are viruses that are able to infect bacteria. And the reason why they use these is because bacteriophages are really just made up of proteins and DNA. And since the question that they're wondering is it DNA or is it proteins that is holding this genetic information, bacteriophages are a really good vessel because that is all they're made of. And so in this particular experiment, they tagged DNA with radioactive phosphorus. This is because DNA is made up of a lot of phosphorus. and they tagged proteins with radioactive sulfur because proteins have a lot of sulfur. And so what they found was when they were tracking both of these different materials, they saw that it was actually the tagged DNA that was getting infected into the bacteria. So that was what was passing through. This led them to go ahead and confirm what Avery found, which was that DNA is the transforming principle. DNA is what is carrying the genetic information. And then the last set of scientists that we talked about was Watson and Crick. They were the ones who discovered the 3D structure of DNA. So this idea that DNA is made up of a double helix and that sort of thing. So those are the main scientists to focus on. Again, make sure that you understand who did what. And now when we talk about DNA and what that structure looks like, DNA is essentially just a chain of nucleotides. So I've drawn a DNA nucleotide here. And so what we see is that in this particular nucleotide, we have a phosphate group, a deoxyribose sugar, and a base. Now, the only thing that is going to differ between different nucleotides is this base. So in DNA, we have 4 options of what the base could be, which is adenine, thymine, cytosine, or guanine. Those are the 4 options. This is the only part of a nucleotide that is going to change. The deoxyribose sugar is going to be the same every time, and that phosphate is going to be the same every time. It is the base that is changing. And so another thing that we kind of mentioned was Chargaff's rules. So this becomes very important when we're thinking of DNA replication and trying to figure out what a complementary sequence would look like. And so those rules are summarized here. A will pair with T and C will pair with G. With RNA, however, which we'll talk a little bit more when we're thinking of transcription, translation, RNA has a very, very similar structure. It has a phosphate group. It has a sugar as well, but in RNA, it has a ribose sugar, and it also has some bases that look a little different than what we see in DNA. So the bases that are available in RNA are adenine, uracil, cytosine, and guanine. So kind of the big overarching difference between RNA and DNA is that RNA uses U instead of T. So this rule becomes very important when we start talking about transcription. So now let's get into kind of the meat of what we have learned in this unit. So this idea of the central dogma, or if you will, kind of the map of how information gets passed along. So we're going to start with this first kind of loop up here. So DNA to DNA. That is the process known as replication. And there's 2 pretty simple steps to this to kind of summarize it. The first step is that the DNA has to unwind and open up. And then the second step involves DNA polymerase. So DNA polymerase is the enzyme that will come in and add nucleotides to be able to make that replicated strand. And so what we see in replication is that we actually call this a semi-conservative process, because in each strand of DNA after replication, one half of the strand is going to be old DNA or template DNA, and the other half of the strand is newly synthesized. So that's all that semi-conservative means. And so once we have DNA and we're replicating in that cell cycle, since this happens during the S phase, replication will only happen one time with each cell cycle. So that is DNA, making more DNA. Then we can move on to transcription. So that is where we take DNA and make RNA. So transcription is very similar to DNA replication. The first step being that DNA needs to unwind. And then the second step involves a different enzyme. So in transcription, we use RNA polymerase to add nucleotides to create a complementary RNA strand to whatever DNA is being read. So with transcription, different from replication, which can only happen one time in the cell, transcription can actually happen repeatedly in the cell cycle. So it'll happen pretty much as we need it to if we are needing to make proteins from a specific gene. So now that we've made RNA, so we have our single-stranded RNA. Before we move on to translation, that messenger RNA that we use in translation needs to undergo some type of processing. So messenger RNA processing has two overarching steps. Introns are removed, and a cap and tail is added. So introns are these non-coding sequences. If a sequence is non-coding, all that is saying is that it won't code for a specific protein. So it's essentially useless information. So we'll want to remove that, add the cap and the tail, and now we have resulted in a fully processed piece of messenger RNA, a fully processed strand. So all of this is happening. So replication, transcription, messenger RNA processing. This is all happening in the nucleus. And then we will actually move into the cytoplasm when we undergo translation. So we'll talk about the steps of translation below, but translation is essentially taking a single strand of messenger RNA and using that as instructions to make a protein. So before we get into the actual steps of this, let's review some concepts from this. So here is a piece of messenger RNA that has been processed. Each set of 3 nucleotides, or 3 letters, however you want to phrase them as, is referred to as a codon. So a codon is a set of 3 nucleotides on a messenger RNA strand. So we have one codon here, and then this is also a codon. So these codons code for specific amino acids. There's a big chart that we talked about in your PowerPoints that you could refer to, but essentially each of these codons will correspond to a specific amino acid that we want to make. And these codons are going to be read by a molecule called tRNA, or transfer RNA. So I've drawn out a tRNA molecule here. The tRNA molecule is going to read the messenger RNA, interpret the instructions, and build the protein that we are wanting to make. So a tRNA molecule has 2 components. It holds a specific amino acid that it codes for, and it has an anticodon. This anticodon is what is going to read the messenger RNA codon. So now let's get into the steps of translation. You can ignore this figure. So For the steps of translation, the first thing that needs to happen is that initiation phase. So a tRNA needs to recognize a start codon. We have one start codon. This is not a sequence that you would need to remember, but just for reference and to make the image a little more easy to understand. The start codon is AUG. And so a tRNA that has the complementary anticodon sequence will come in with its corresponding amino acid. So it's found the start site. It will recruit the ribosome. So we have a large subunit at the top of the ribosome, and we have a small subunit at the bottom. So this tRNA will recruit this ribosome and say, okay, now we can start translating this messenger RNA strand to make our protein. So once that transfer RNA binds, the next transfer RNA for the next codon, which would be these 3 nucleotides, can come in and bind. So we see that the anticodon is complementary to the codon that it is reading. And so once it's able to bind and bring in the correct amino acid, what essentially is happening is the amino acid from that first tRNA will be moved and form a peptide bond with the new amino acid that came in. So this reaction and this peptide bond is being catalyzed by this large subunit. The small subunit of the ribosome is functioning to just hold our messenger RNA in place. That way we can read it and make sure that it's easier to read. So once that amino acid can form the peptide bond, this empty tRNA will leave. It's done its job. It's given us the amino acid. And the next tRNA can come in. And so the same thing will essentially happen where these 2 amino acids will be able to join this one. And this process will just keep repeating itself and keep moving down the messenger RNA molecule until we reach a stop codon. When we reach a stop codon, the messenger RNA can be released and the ribosome and everything will disassemble until we want to undergo translation once again. So we'll end up resulting with this chain of amino acids that make up a polypeptide or a protein, whichever terminology is more comfortable for you. So those are the 3 big steps or processes that we covered. So now let's go ahead and move into this idea of gene expression and regulation. So we talked about it first with prokaryotes. So this is the idea of the lac operon. So I have both images here. So we'll just walk through these really quickly. So essentially what we are saying is with gene expression and regulation, we don't want to waste our energy making proteins that we don't actually need. And so for the lac operon, we are making enzymes that will digest lactose. That's what the lac operon is doing. And so we have to think about what conditions are present. So in this first example, if we are in an environment that has lactose, it would make sense that we would want to go ahead and create the gene. We would want to go ahead and transcribe the genes that would create the enzymes to digest lactose because it is present in the environment. So in this particular case, since we do want to go ahead and go forward with transcription, what will happen is the lactose that is present in the environment will bind to a repressor, kick it off of the operator, and allow that RNA polymerase to go through transcription. With an environment that does not have lactose, we wouldn't need to make those enzymes. And so we wouldn't want to undergo transcription. So what happens is that repressor will bind to the operator, blocking RNA polymerase from transcribing. And so a couple of definitions that we kind of talked about in these two scenarios, just to kind of clarify. A promoter is simply just a part of the DNA that is telling RNA polymerase where to start transcription, while an operator is kind of acting as this on and off switch. And so in the lac operon specifically, a repressor is bound. You can think of it as being on the off switch. If the repressor is not bound, then transcription is turned on. We also talked about gene expression and regulation in eukaryotes. And so this was the image that we talked about, and similar to prokaryotes, eukaryotes have a Tata box promoter. That's just the promoter that is specific to eukaryotes. And eukaryotes also have things called enhancers or silencers. So these can affect the rate of transcription. So enhancers would speed up transcription, while silencers would slow it down, just depending on what the needs of the cell are at that time. We also talked about mutations, again, more definition based, but there are images for each of these that can help give you a good visualization of them. But there are essentially two kinds of mutations. So there are those that can affect a single gene, and there are those that will affect an entire chromosome. And so the examples that we talked about of mutations that affect a single gene are point mutations, which are when we essentially are seeing an exchange of a nucleotide. So in a particular sequence where there was an adenine, it underwent a mutation, and now we see a cytosine. So we went from having an A to a C. So it's just exchanging or changing whatever base is present in that location. We also talked about frame shift mutations. So you can have a frame shift insertion where a nucleotide is added, or you can have a frame shift deletion where a nucleotide is removed. So they're called this, and the name should really help you distinguish this, but a frame shift mutation is shifting the reading frame. So if we're adding or if we're taking away a nucleotide, that could completely change what the codon would be read as. And so that will change the resulting protein and just cause a pretty big change in that particular example, if it's in a coding region. For the mutations that affect entire chromosomes, so we talked about gene duplication, which is this idea of unequal crossing over, and translocation, which is when gene segments of one chromosome exchange with gene segments of a completely different chromosome. So these are going to affect the entire chromosome and will likely lead to massive changes in phenotype. But we could have when we are talking about these mutations that affect a single gene, we could have what we call a silent mutation, which is when a mutation occurs in a non-coding region. So essentially, the effects of that mutation are never seen because we don't code for that particular region. So if the mutation happens in body cells have no part in fertilization or sexual reproduction, and so those mutations would not be able to be passed off into offspring. However, if these mutations happen in sex cells, they can be passed on to offspring because it would happen in the gametes. The gametes would come together in fertilization and pass down whatever this mutation is. And mutations can be caused by a couple of different things. These are the examples that we talked about in class that you should keep in mind, but they could be caused by replication errors that our DNA polymerase maybe didn't catch. It could also be caused by UV rays, chemicals, things of that nature, different mutagens, or even drugs that we use in cancer treatment. So that summarizes Chapter 8. This summarizes the scope that you should understand everything, the content that will be covered on the test. And so now we'll go ahead and move into chapter 9. So I very much simplified it. These are the definitions you need to know. And so I'm not going to go ahead and read all of these. You can refer back to these if you want to make a small Quizlet or something. The only thing I want to just make sure that I touch on is the gel electrophoresis. So there is a similar question that we did in the in-class question, so you can use that as practice. But essentially what could happen is I could give you an image just like this one, where it is a rectangle with different lines on it. And so with gel electrophoresis, the whole concept of this is we are separating DNA fragments by size. So we take our DNA, we add restriction enzymes to cut up the DNA, and we run it through the gel. So the number one rule in being able to understand gel electrophoresis is the smaller fragments will move down farthest. So if I were to ask you which fragment, A, B, C, or D, is the smallest fragment, it would be the one that moved the farthest down. So that would correspond to D. If I asked you for the largest fragment, the larger the fragment is, it's the less likely it will be able to move down. So that would correspond to letter A. You can also think of these in comparison to one another. So even though we know D is the smallest because out of all 4 of these bands, it traveled down the furthest, we could also say that B is smaller than A, even though B is pretty high up on the gel, since it has traveled further down than A, we can kind of make that relative comparison. And then you can just go ahead and review the different vocabulary. Again, I would recommend making a Quizlet for these. The format of the test is going to be the same as it has been, so 15 multiple choice, 10 matching, which a lot will come from these different vocabulary. And then there'll be 2 short answers. Both of the short answers are actually going to come from chapter 8, just because conceptually we didn't really go into any detail with chapter 9. So just kind of keep that in mind. And so if there are any questions, please let me know either through email, in class, whatever you prefer. You're also welcome to come in during A-Lab or pretty much anytime you have free that you want to walk through things, or just make sure that you have the correct understanding of either concepts or the correct understanding of the expectations for the test. But other than that, watch this multiple times. That would be my recommendation. If there are things as you rewatch this that still don't make sense, focus on those PowerPoints in particular. If something was not mentioned, especially within chapter 9, then that's not going to be covered on the test. Another recommendation is, after you've gone through this a bunch and you feel confident with the material, go ahead and go back and rework those chapter 8 and chapter 9 in-class discussion questions. Just like the last test, you will likely see some of those again. So make sure that you look at those, because we've gone over them before, so I would hate for someone to get those wrong the second time around. But other than that, happy studying, and let me know if I can be of any help.